Thanks very much for the invitation and the kind introduction. It was also very nice that I got to coordinate this with May Day yesterday. So we got here yesterday morning and were able to walk around the city and see all the white hats and all the celebration. It was very, very nice, very pleasant. So I thought what I would do today is give some introduction on depression, and then I'll get into a lot of the work that we've done on stress, and in particular on the mechanism of action of rapid-acting antidepressants. Some of this work may be familiar to you. The impact of depression on the population, higher rates, incidence in women compared to men, major economic impact over time, and the available treatments that we have mostly require many weeks or months and have some limited efficacy. And it's surprising in this day of modern medicine that we still don't really understand the neurobiology of depression or exactly how the treatments that we have are working. So work that has been done in our lab, but also a lot of work here done by Eero Castren as well as Tomi have focused on this idea that there's a neurotrophic component that with stress and depression there's a loss of neurotrophic support, and that antidepressant treatments can reverse that neurotrophic loss. And just in terms of some clinical background, some of the first evidence for this hypothesis was based on evidence, brain imaging evidence, that the size of some brain regions implicated in depression, like the hippocampus and the prefrontal cortex, undergo an atrophy that's associated with depression. And then there's some post-mortem data showing loss of some glia cells has been well established, but also a few studies like this actually looking at synapse number in brains of depressed patients and showing a significant loss of synapses here in the dorsolateral prefrontal cortex. So then, of course, in the animal models, we're able to go into much greater detail of what underlies the structural changes that we are seeing and that contribute to these MRI and postmortem studies in depressed patients. So one of the models we use is a chronic unpredictable stress model, also chronic restraint stress. This is a study done in collaboration with George Agajanian, where they record from pyramidal neurons in the prefrontal cortex, and then they fill those cells while they're recording, and then we can go back and image those cells and actually measure dendrite length and branch number and spine number. This is a relatively mild 7 days of restraint, 45 minutes a day, and you see this very robust retraction of the apical dendrites. And as I mentioned, if you get down to a higher level of resolution, you can see the spine synapses that decorate these dendrites, and that there's a reduction also in the number of synaptic contacts after chronic stress exposure. So, you think about this, maybe the mouse wouldn't think it's a mild stress, 7 days of restraint stress. But when you think about the stress we all have in our lives, that 7 days of some type of stressor is not a huge factor. And the idea that could be contributing to this volumetric change in depression could be related to the therapeutic or the syndrome or the symptoms of depression, not only for depression, but maybe for other psychiatric illnesses as well. It's something we've been interested in looking at and studying. So what I thought I would do first is just give you a little bit of an overview of some of the studies that we've done, trying to understand the mechanisms and pathways that contribute to the atrophy that we're seeing with stress. And then I'll get into quite a bit more detail with some of the more recent work. So here's just a couple of areas that we've been interested in. This mTORC signaling pathway is one that has been studied in long-term potentiation, a cellular model of learning and memory. It's a protein synthesis activity-dependent component of L2P. And mTORC is found in all parts of the cells and all types of cells. but it's located in the right place. It's in dendrites and synapses in neurons. So I'll show you one just overview of the response at some studies we did on mTOR. And then just, again, very brief overview on the role of microglia, which have also been implicated in stress response and even in atrophy of neurons in response to stress. So again, just a quick overview, and just also to mention that there's been a lot of work looking at BDNF, one of the major neurotrophic factors in the brain, that is known to be very significantly downregulated in the prefrontal cortex and the hippocampus in response to chronic stress. So we know that that component has already been reduced in these chronic stress models, and there's evidence from postmortem studies of a loss of BDNF as well. BDNF is one of the signaling molecules that leads to activation of mTOR. And what we were able to find and show is that there's another negative modulator of mTOR that's upregulated by chronic stress that inhibits mTOR signaling and inhibits synapse formation, or reduces or contributes to the loss of these synapses in the stress condition. So that if we block RED1 signaling, we block the effect of chronic stress on synapse formation and antidepressant-related behavioral deficits that are seen in these models. So I think that in combination with BDNF reduction is one of the major signaling pathways contributing to the stress response. And then in the case of microglia, many of you may have heard and been reading about inflammation, not only in the brain, but basically every area of medicine is, I think, interested in how inflammation is impacting different systems. And in the brain, it's known that the microglia, which are the resident microimmuno cells, that they, under normal conditions, which can show sort of what we have here on the right side with antidepressant therapy, they actually are located around synapses, and they are involved in the maintenance and function of those synapses. And with different kinds of immune activation, you can see activation of these microglia. They take on this different shape, and they actually start to engulf the spine synapses along those dendrites. And what we've been able to find with stress is that there's an increase in activation of the microglia, and an increase in the number of microglia that have engulfments of neurons that are labeled to show that gives us a way to show engulfment by the microglia in response to stress. And that there are factors that actually are released from the neurons that are regulating these microglia. And if we block those factors, we can block, again, some of the effects of stress exposure. So that's kind of the background on some of the stress studies, that we have not only these pathways that are acting on BDNF and some of the synaptic signaling cascades, but that microglia are also becoming activated. This molecule, choline-stimulating factor 1, seems to be involved, and that they're playing a role in the engulfment and reduction in number and size of the synapses. So if we then go to how antidepressant agents are interacting and involved, again, we have these monoanergic agents like serotonin selective reuptake inhibitors such as fluoxetine that have been widely used for many, many years. Very beautiful evidence, again, from studies here in Eric Castron's lab as well as Tomi, showing that SSRIs can enhance synaptic plasticity of very, very elegant models looking at ocular dominance and extinction of fear conditioning. Some studies that these agents can directly influence spine number and spine density, and a little bit of data that they can block the reduction and the atrophy of neurons in response to chronic stress. But as you may know, these agents typically take weeks or months to produce these effects in these models. Take weeks to months to produce a therapeutic response. Only about a third of patients actually respond to typical antidepressants, up to 2/3, but that can take many years or over a year or two to find the right combination of a monoaminergic agent, maybe in combination with something else. So that leaves, at best, about 1/3 of patients who are considered treatment resistant. So a few years back now, it's actually been more than that, this study was published by John Crystal and colleagues at Yale, showing that ketamine, again, a drug that many of you may know about, is a dissociative anesthetic. It's used in pediatric medicine, veterinary medicine. It's an NMDA receptor antagonist. And what they found was a low dose, 0.5 milligram per kilogram, which is just enough to produce some dissociative psychotomimetic effects. When it's infused over the course of 40 minutes, it then, within a matter of hours, produces this very robust antidepressant response, even in patients who had not responded to other typical antidepressants. And this is just to show that some of those other effects, the dissociative or the psychotomimetic effects linked to the sort of schizophrenic-like response, the dissociative effects, occur very rapidly within the first 60 minutes. It's not until after that, within 2 to 4 hours, that you start to see this therapeutic response, sort of dissociating those early effects from the therapeutic effect. That was sort of a small study. This study from Carlos Zarate was a much larger placebo-controlled study where they found even as early as about 2 hours after that single dose, they saw a therapeutic response that was sustained for about a week. So patients stayed better after a single dose for about a week, and then they do relapse. So there is this relapse problem that occurs after that single dose. But still, and considering that these patients, again, are treatment resistant, and showing that this effect can occur within a matter of hours when all other drugs that we have available take weeks or longer, is really an amazing change and has had an amazing impact on the field. And just a little bit of other clinical data I think people find interesting. It also seems to be very effective for reducing suicidal ideation. Some of these studies done in the ER, patients are brought in and are suicidal. They treat them with ketamine. Their suicide ideation is reversed or reduced, and they can stay better for several days, which is a huge improvement over current clinical interventions for suicidality, which is very difficult to treat. And then this is another aspect. Again, people often ask, well, what about repeat dosing? There have been a few studies like this that have been done, not a lot, but here they show that dosing every other day or for I think 2 or 3 times a week is what they're doing here. People remain better over the repeat dosing. And in one study, they actually found that people stayed better longer after they stopped the dosing. So this does seem to be some enhanced therapeutic response with repeat dosing. And then this, I just wanted to show because this is a stereoisomer, the S stereoisomer of ketamine. It's been developed by Janssen & Johnson as a nasal application. And they show very nicely in some of these early studies that it also produces a rapid antidepressant effect that lasts for several days. And this drug was just approved by the FDA in the USA, so they're now setting up clinics. And so we should see, I think, probably within the next year, that this will become something that's being prescribed much more frequently. The earlier studies with IV were mostly in clinical research trials. So this will be a way to really be able to get this out in a broader type of way. So just putting this in perspective, the field developed the monoaminergic drugs back in the 50s. They started being used during that time. There were some refinements in the SSRIs that helped with the side effect profile, but basically they're the same types of agents that we've had for over 60 years now. A couple of very big failures by the pharmaceutical industry that has really led to most big pharma getting out of psychiatric research because it just was not paying off. The preclinical models were not predictive of what was going to work clinically, and they've basically pulled out for the most part, which is a big problem because we really need better treatments. So when you think of ketamine, it's the first new drug, new mechanism that we've had in many decades, and it's effective very rapidly in these treatment-resistant patients. So what I'll spend the rest of the talk is how this NMDA receptor antagonist is able to produce this rapid and robust antidepressant response. As I mentioned, ketamine is an antagonist in the NMDA receptor. It's an open channel blocker. So that means that typically this channel is closed if the cell is not active. When the cell is depolarized, you remove this magnesium block. That allows ketamine to then drop into the channel and block. And there are other channel blockers, but this ketamine has some somewhat unique properties in how it blocks the channel. There are some other compounds that I'll come back to on here, negative allosteric modulators, some positive allosteric modulators that are being developed. But NMDA receptor drugs are now a very hot area for development for antidepressants, as you could imagine. So one of the things that has been interesting and paradoxical about this NMDA antagonist is that when you inject it, this is a rodent study, you see that within about 30 to 60 minutes, it actually causes this increase in extracellular glutamate in the prefrontal cortex. So how does an antagonist lead to an increase in glutamate? And that we think that that's very important and may be linked to the subsequent therapeutic, synaptic, and therapeutic responses. That's what's shown here. Could this burst of glutamate somehow produce an LTP-like activity-dependent increase in synaptic activity that could be playing a role in reversing the synaptic losses that you see with chronic stress exposure? So again, this is work done with George Agajanian's laboratory, where they were patching onto layer 5 cells in the prefrontal cortex, filling those cells. So not only do we have functional data on the synapses, but then we can also do these structural studies. And largely because of the logistics of doing the slice preparation, we typically administer ketamine, wait 24 hours, make the slice, stabilize the slice, and do all the physiology and the labeling. So this is 24 hours after a single dose. And we're looking at basal rates, not much change in basal rate activity in the prior ketamine dosed animal. We see in response to serotonin and hypocretin, very significant increase in particularly the amplitude, but also the frequency of these EPSCs. And that's what's shown down here, response to both hypocretin and 5-HT. And then again, if we go back and look at spine density on these cells, we see a significant upregulation of their number of spines at both a distal as well as proximal level of the dendrite. So we see an increase in both number as well as the function of dendrites within a relatively short period of time after that single dose of ketamine. And then to get at this more acute phase within a matter of hours after the dosing, we've looked at a number of synaptic proteins, including synapsin-1, PSD95, and BluR1. And now we can administer ketamine and collect the tissue within very discrete time points, again going out to 72 hours here. But the important thing we see, within 2 hours we get a significant increase in synaptic proteins. So we think that we're getting new synapses or at least new synaptic function within a time frame, I think I have it here, that overlaps pretty nicely with the time course for the therapeutic response. This is from, again, that Zarate study where they see a therapeutic response within a couple of hours, which is when we see that synaptic change. So it's only an association at this point, but we think that that's at least consistent with that hypothesis. And then going one other study to sort of get at this, the relevance of these synaptic changes, we've looked at this also in the context of one of these chronic stress models. which serves a couple purposes. We can test the validity of a rapid-acting agent, the behavioral effects of a rapid-acting agent. Because in this stress model, when you expose animals to chronic stress over the course of 3 weeks, it produces an anhedonic state that you can measure by looking at the amount of a sweetened solution the animal will take. A typical antidepressant you would have to administer for 3 weeks before you would see a reversal of that behavioral deficit. So we can test ketamine to see if it produces a rapid response. And then we can also see if it reverses that synaptic spine loss that occurs with the chronic stress exposure. And that's basically what we find, that single dose of ketamine is capable of reversing the spine deficit as well as the function of those spines, looking again at response to EPSC, response to serotonin and hypocretin. And also in the behavioral model, this sucrose preference test, you see this reduction with chronic stress that's again reversed with ketamine exposure. We also looked in it's more of a model of anxiety, novelty suppressed feeding. If you put a mouse or a rat in an open field, it doesn't want to go to the open field, which is usually where some prey animal will come and get it. If you give the animal exposed to chronic stress, it will increase the latency to go to the middle of the field, and ketamine reverses behavior in that. A single dose of ketamine reverses that behavior as well. So I probably don't need to go over this at this point, but this is sort of our simple model that stress is leading to a reduction in spine synapses and prefrontal cortex. We think through reductions in BDNF and BDNF signaling, maybe also microglia playing a role, in that these rapid agents are able to very rapidly come in and reverse that synaptic loss. And we think that that's contributing to the antidepressant behavioral effect and possibly to the therapeutic response as well. Time course is consistent with that. So what I'll do now is sort of give you an update where we are at this point. We've been focusing on a number of different areas that we think are important to further understand how ketamine is acting. And going back to this glutamate burst, this sort of paradoxical glutamate burst, one theory is that that's occurring through blockade of NMDA receptors on GABA neurons. So you remove that GABA inhibition and you get this burst of glutamate by the excitatory neurons. So that's one project I'll talk to you about. Another one is looking beyond BDNF at a second trophic factor, vascular endothelial growth factor. And I'll show you some data there. And then a third area that we've become interested in is trying to understand if direct activation of these pyramidal cells can reproduce the effect of ketamine. And then if so, are there subtypes of pyramidal cells that might be important for that response? Okay, so the first project, again, going to this idea, how does this NMD antagonist produce this paradoxical burst of glutamate? There are some theories about why this might be more important than possibly a direct effect at NMDA receptors on the postsynaptic cell. And we've sort of referred to one of those, and that is that ketamine is an open channel blocker, so that it has to have an active neuron, a neuron that's firing to remove that chloride, that magnesium block. And so these GABAergic interneurons tend to be more tonic firing to maintain sort of a lower level of glutamate activity. So they may be the GABA neurons may be more sensitive than the glutamatergic neurons because of that tonic firing activity. A couple of the things that it's important for this project to sort of take into account. We know the prefrontal cortex, particularly the medial prefrontal cortex, is an important region. If we infuse ketamine into the medial prefrontal cortex, it produces the same antidepressant effects as a systemic dose. If we put a neuronal silencing agent into the medial prefrontal cortex, it also blocks the effects of systemic ketamine. So medial PFC, we wouldn't say it's the only region, but it's we think one of the key regions for producing a ketamine response. And then we're going to target in these studies the GLUN2B subunit of the NMDA receptor, and that's based largely on some data with GLUN2B selective negative allosteric modulators like this CP101606, which has been shown to produce a similar type of rapid antidepressant effect, similar to what has been observed with ketamine. There are also some other compounds, GLUN2B selective NAMs, that have been tested in rodent models. So basically, the approach that we're going to take is to target NMDA receptors either on the GABA neurons or on the pyramidal excitatory neurons, and see if we can selectively knock down or block the effect of ketamine to test this direct hypothesis of whether we're having effects directly on pyramidal cells or indirect, whether ketamine is acting indirectly through the GABA interneurons. And here's what we're doing. We're using a viral vector that's a Crelox selective system to express a GLUN2B shRNA, and we're driving that under either a promoter for excitatory neurons or a promoter for inhibitory neurons. For excitatory neurons, it's CAM kinase 2A. For inhibitory, we're using GAD1. I'll show you a little bit of data with either somatostatin or parvalbumin as well. And on the top here basically is sort of validating the model. If we express this construct with Cre in primary neurons, you can see reduction of GLUN2B. If we do this viral infusion into rodents in the medial prefrontal cortex, make slices, record from the neurons, we can see a loss or reduction in NMDA response in both the CAM kinase 2A mice, no effect on the AMPA response. In the somatostatin Cre mice, also a reduction in NMDA, but no effect on AMPA response. So here's some of the data. We are just submitting this data now. So it'll be interesting to hear your feedback. And so we're, again, starting out infusing into either the GAD1 to knock down GLUN2B in the GABA neurons, or in CAM kinase 2A to knock down in the excitatory neurons. We do some initial testing on the animals to see if there's any baseline differences. We do see in our forced swim test, if you don't know that, very well-established old model of testing antidepressants. It's putting an animal in a container of water. You measure how long it will swim or struggle to stay above water. It eventually gives up and will float. And so you measure the time it swims or the time of immobility. Typical antidepressants will reduce the amount of immobility. So it's supposedly a model of helplessness. That's been debated, but it seems to be at least a good marker or good test of antidepressants and typical antidepressants. And so what we find in the pre-swim, you give a couple of exposures in the pre-swim. We see already in the GAD1 mice, where we have GLUN2B knocked down in the GABA neurons, that there's an antidepressant-like effect, a reduction in immobility, which you might predict would be acting somewhat like ketamine blocking the NMDA receptor on those neurons. No effect in open field, either center time, which is, again, measure of anxiety, how long the animal will stay in the center, or the total distance traveled. So we shouldn't have any problems of interpretation with changes in total locomotor activity. If we then look in the test, forced swim test itself, again, ketamine in the control animals receiving just the vector without the shRNA, we see the ketamine antidepressant decrease in immobility. Again, now we see even a more robust antidepressant-like effect in the GAD1 mice, and ketamine has no further effect. So there's an occlusion of the ketamine response under these conditions. What we see in the novelty-suppressed feeding test, again, this is more a test of anxiety that's responsive to typical antidepressants. Again, a reduction in latency to feed in the middle in the ketamine-treated animals, and some occlusion and some blocks. So it's sort of a partial blocks, partial occlusion in response to knockdown in the GAD1 Cree mice. No effect on home cage feeding, which is a control for the novelty breastfeeding. And then by comparison in the CAM kinase 2, where we're knocking down in the excitatory pyramidal neurons in the prefrontal cortex, ketamine response in the controls, and almost no difference in the CAM kinase 2 knockdown animals. You can see almost no difference, or no blockade and no occlusion. And basically the same thing in the novelty suppressed feeding. So this is the first data we have that would indicate that this indirect hypothesis that ketamine is acting on NMDA on the inhibitory neurons, but not the excitatory neurons, is likely the case. And then I just show you a little bit of data that we've taken this to the next step to look at subtypes of GABAergic neurons. Two major classes, there are subclasses and other types as well. These are the two major types of inhibitory neurons in the brain that express either somatostatin or parvalbumin. The somatostatin or Martinati cells are slow spiking. They tend to innervate more dendritic terminals. They tend to innervate the terminals and control flow into the cells. Parvalbumin neurons tend to innervate cell bodies, are involved more in the output. So we're testing whether or not ketamine can act on one or both of these. I'll show you some data with somatostatin, but we've seen very similar effects with parvalbumin. And it looks like both cell types are actually contributing to the ketamine response. So very similar to the studies I just showed you, except now we're using a somatostatin Cree mouse, Cree recombinase mouse. So we're expressing the shRNA to knock down GLUN2B selectively in the somatostatin cells. And what we did here, we compared male and female, saw a slightly bigger effect on pre-swim in the males compared to the females. And at least in this pre-swim test, but not necessarily in some of the other behaviors. I'm just showing you the male data here. The female data looks pretty much the same after we give ketamine. Again, we see this sort of partial occlusion to the ketamine response. Again, here's our control response, the knockdown in SST by itself, and no further effect of ketamine in those animals. Here's probably one of the cleaner data sets that we have. Again, the novelty suppressed feeding, we get this decrease to ketamine in the control mice and a complete blockade of that response in the SST knockdown animals. So again, I think pretty consistent with definitely these neurons are contributing. They're knocking down NMDA produces some effect that's similar to the ketamine response, and also in some cases is blocking completely the ketamine response. This we were particularly interested in the somatostatin neurons. So we also did a genetic deletion of GLUN2B in the somatostatin neurons using, again, an approach where we're crossing these Cree recombinase mice, in this case with a Cree-lox GRIN2B mouse, to produce deletions selectively in the somatostatin neurons. And then basically doing the same types of behavioral studies. Interesting, again, in the pre-swim, we see this effect in the males, but not the females. So it's sort of similar to what we found with the viral knockdown. No effect in open field. Males, we also do another test now. We've started looking at more, it's a measure of motivation and reward, where a male will like to spend more time with a cotton swab with female urine. So you can only do this in the males. But it's still interesting to look at. And we found that ketamine produces an increase in the time that the animal will spend. So it's sort of a measure that you're getting an increase in motivation and reward. We find in the males that with knockdown of GLUN2B and somatostatin, or knockout in this case, that we're getting a ketamine-like effect, or already have this increase in time spent with the female urine. And then if we then treat the animals with ketamine, giving a single dose during the behavioral tests, here we're again seeing more of a blockade of the response, where in the forced swim, again, our typical response to ketamine, some small effect in the knockout alone, but a complete blockade of the ketamine response, and somewhat similar, although maybe slight occlusion in the novelty suppressed feeding test. So overall, I think the data, I'm not going to show you the data with the parvalbumin, but it looks similar to what I showed you with the somatostatin. We think that, again, both somatostatin and parvalbumin neurons are contributing to the ketamine response. Inhibiting both of those cell types is leading to this, we think, this burst of glutamate that then drives the synaptic activity in the pyramidal cells. Okay, and then the next part I would just thought it would be interesting for this group. I know you guys have done studies on growth factors. We've been interested for some time in BDNF itself. Became interested in this vascular endothelial growth factor probably back in the 2000s, where we found that we and others had found that seizures, like an electroconvulsive seizure, produces a big increase in BDNF expression. And when we looked at other factors, we found a similar response with VEGF. You get this big, robust increase in VEGF expression. And even though it's called vascular endothelial growth factor, it's a pleiotropic growth factor that is expressed by neurons, it's expressed by glia. Receptors are expressed in neurons and glia, so it's a very potent neurotrophic factor as well as an endothelial factor. And what I'm going to show you, we're using techniques that are similar to what we had shown for the studies looking at GLUN2B, either using a genetic deletion of VEGF itself, or it's one of its primary receptors, FLK1, or we're also doing this viral shRNA knockdown for the receptor. So again, here we're looking specifically whether or not VEGF in the principal neurons is playing an important role in the ketamine response. So again, we're crossing with our CAM kinase 2A Cre recombinase mouse with a floxed VEGF to delete VEGF from the neurons specifically. And that's probably, that idea would be related again to the fact that when you depolarize and activate these neurons, you're getting not only synthesis of BDNF and VEGF, but you also get release of those growth factors that have then effects on the same pyramidal cells or on other cells around. So again, when we do looking at response, and again in forced swim and novelty suppressed feeding, ketamine produces again this decrease in immobility. In this case, we see a complete blockade in the neuronal VEGF deletion mice. Similar reduction in latency to feed that's blocked in novelty suppressed feeding. I told you about this, the female urine test. Again, the antidepressant effect is blocked by the deletion. And the tail suspension is basically another helpless-like. It's sort of like the immobility for swim immobility. You get a decrease in immobility that again is blocked in the VEGF knockouts. We have similar genetic deletion data, but I thought I would just show you this viral deletion because it also gets at the question of what brain region is important. With the genetic deletion, we're getting deletion throughout the entire brain. Here we're able to, as I showed with the GLUN2B, we're able to focus that knockdown to the medial prefrontal cortex, again, this region that we think is important to the ketamine response. So again, this is just expressing the shRNA. In this case, we're expressing in the CAM kinase 2 to drive expression. So now we think this is happening, or it's important to the pyramidal cells specifically, so we're knocking down there. And again, you can see that when we knock down the receptor FLK1 in the pyramidal cells, we see a blockade in these various models for swim, the urine test, knowledge breastfeeding. I think I just had a couple more slides. This is testing whether or not VEGF release might be important. So when we knock it down, it's hard to tell whether it's having some prolonged, the knockdown is having, or the knockout is having some prolonged effect on the function of those neurons. So here we're infusing a VEGF antibody directly into the medial prefrontal cortex just prior to ketamine administration. And then any VEGF that's released would be bound by the antibody and be neutralized. You can see basically the same kind of results we were seeing with the VEGF deletion, that you're getting a blockade in the forced swim, the novel breastfeeding, and this urine test. And just, I think, one or two more here, testing whether VEGF itself is sufficient. So those studies will demonstrate that VEGF is required for the ketamine response. We were also interested to see if VEGF could sort of mimic the effects of ketamine. And we find very low doses, 5 nanograms infused into the medial prefrontal cortex produces a very robust antidepressant effect, in these various models. And I think I just have this one last slide on the VEGF part of this that I wanted to share, because it gets back to the looking at these spine synapses. Here we're looking at not using the labeling, but actually just Golgi staining prefrontal cortex, then looking at spine density at a couple of different levels, either again proximal or distal, 1 or 2, and also in the infralimbic cortex as well as the prelimbic cortex. Again, you see with ketamine an increase in spine density that's blocked at both levels in the infralimbic as well as in the prelimbic cortex. It looks like not only BDNF contributes to the structural spine changes, but that VEGF is also contributing. So that is just a quick, brief summary that VEGF as well as BDNF, we think, release are contributing to the synaptic changes that are occurring in response to ketamine administration. And this is the last project. This is something that we finished up just recently. Again, we started out wondering to test this direct versus indirect hypothesis and whether or not this is an activity-dependent effect. Because there was some data that from some labs showing that you could produce some synaptic homeostasis at higher doses of ketamine that apparently would only work in the absence of activity. So we wanted to test whether activity could substitute for the ketamine antidepressant response. And then test, in a second project, if we could determine what types of principal neurons in the prefrontal cortex are playing a role in the ketamine response. So we've been using some optogenetic approaches. This is the first study, just expressing channel rhodopsin under the control of the CAMP kinase promoter to express in all pyramidal cells in the prefrontal cortex. And we're doing this a little bit differently. We want to basically try to mimic what's happening with ketamine. So we're giving a 1-hour stimulation, 1 minute on, 1 minute off. I think we're using 10 hertz in this study. But we're not doing the behavior during the stimulation. We're waiting 24 hours. So we're looking at, again, the subsequent effect on behavior of the synaptic changes that are occurring during this previous 1-hour stimulation. And you can see in these animals that receive this optogenic stimulation, then are tested over the course of the next 2.5 weeks, that we get a very robust antidepressant effect, again, in the forced swim, novelty breastfeeding, 5 days out. Even in sucrose preference, typically you don't see an effect in naive animals, but we saw some increase here only in animals that were getting bilateral stimulation, not with the unilateral stimulation. And even 17 days later, we still were getting an antidepressant response after this stimulation. So definitely the direct activation of these neurons mimics the effect and produces what looks like an even longer lasting antidepressant response. Looking at the spines, basically we see a very similar kind of increase in spine density and function that I had described with ketamine itself, both an increase in density. We've also looked at spine head diameter, which is a measure of the number of functional spines, the mushroom spines being more functional. And then again, also in the response, the frequency response to serotonin and hypocretin. So, we're more molecular biologists, biochemists. We take chunks of tissue and grind it up, and it's becoming more and more clear. I showed you studies of subtypes of the GABA neurons. There are also now more and more evidence that there are many types of principal neurons in the prefrontal cortex. We sort of started this by, we had a chance to use these mice that are dopamine D1 or dopamine D2 expressing mice driving Cre recombinase. Some people in our group had them. We're studying neurons and responses in the striatum, which have a lot of these D1 and D2. But these D1 and D2 are also expressed in subtypes of principal neurons in the cortex, including in the prefrontal cortex. And basically, these DRD1s, they have a different morphology, somewhat smaller arbor compared to the D2 expressing. They differ in some of the physiological properties. The voltage sag is lacking here, whereas the D2s do. So we can identify these when we record from them. We know whether we have one type or the other. I won't show you any data, but we do see some differences in the physiological responses in these neurons as well. And then we're using, again, D1 Cre or D2 Cre mice to drive expression of channel rhodopsin, specifically in one cell type or the other. And so I'm just going to show you data from D1 or D2. And the take-home story is that that says D2 up there, that's supposed to be D1. This slide is D1. That when we overexpress channel rhodopsin in the D1 neurons and photoactivate, again, 24 hours ahead of the behavioral studies, that's sufficient to produce the same effect that we saw with the CAMP kinase. We see an antidepressant effect in forced swim, also in an elevated plus maze, another model of anxiety. No effect in this sucrose preference. That is one difference. But then also in the novel breastfeeding, and even at a later time point in that forced swim test. And then by contrast, we do the same thing with D2 mice, activating D2 neurons. No effect in forced swim. And actually, it looks like we get a little bit of an increase in anxiety in the elevated plus maze. And I think I just had a couple of other slides here I was going to share. Here, testing whether or not these neurons are involved in the ketamine response. We've done this both optogenetically with a halorhodopsin, inhibitory opsin, but we found that we get a better inhibition using a DREAD approach. This is this GI-coupled DREAD that you can overexpress and then activate when you administer a synthetic analog of clozapine, CNO, to the animal. Will activate, in this case, it will bind to that inhibitory DREAD, drive inhibition right after we give ketamine. And when we do that, we can see that when we block the activity of the D1 neurons, it blocks the effects of ketamine in the forced swim as well as in the novelty breastfeeding. So these D1s are required also for a response to ketamine. And then I just had one or two other slides I can wrap up pretty quickly. One piece of data that's overlooked, I showed you this glutamate extracellular data that showing that ketamine produces this response. In the same paper, they show that dopamine, extracellular dopamine, is also increased with a very similar time course. It hasn't received as much attention by most of the field. And when we started these studies, we weren't really interested in dopamine per se, but we thought, given this data, maybe we should look at dopamine. And so we've done a couple studies now using some pharmacological approaches, infusing either a D1 agonist into the prefrontal cortex or an antagonist in combination with ketamine. And the agonist produces some mild antidepressant effect in forced swim and novelty-suppressed feeding. And the antagonist, D1 antagonism, blocks the effect of ketamine. So it looks like you need to have some dopamine, extracellular dopamine there to see the full ketamine behavioral antidepressant effect. I think I will just skip this because we're running out of time. But we're starting to look at some of the downstream circuits, particularly the amygdala and ventral hippocampus in these responses as well. So this is what we think is happening, that initially ketamine is binding, blocking NMDA on the GABA neurons to produce this disinhibition, glutamate burst, that the D1 type of neurons seem to be most important to the response. That activation results in voltage-dependent release of BDNF and we think VEGF as well, playing a role, activating multiple pathways. We certainly are certainly interested in this mTORC1 pathway, but there are other pathways that have also been implicated to increase spine synapse number and function and output on those neurons. And this has really become, after a period where it looked like we were just running out of options for new treatments for depression, has really become a very big area of interest in research and drug development, which is fantastic. And if you come to the thesis defense tomorrow by Samuel, you'll hear him talk about a very novel compound, nitrous oxide. So I'll leave you with that, put up the acknowledgments of people who did the work and as well as funding. Thanks for your attention.